And I'm going to put the link to the posters into the chat in case you have not looked at them yet. We're going to have 4 posters, and each poster, the author is going to have about 10 minutes to present them. I think we have some live presenters and 1 video for the posters. And again, during this session, the code of conduct applies. So I hope you're all familiar with that by now. Questions here, we can have them live or put them in the chat. I will try to monitor the chat as good as possible, because I mean, if there's a lot of posts, it might be harder, but we probably can have the questions live. We have a couple more minutes before we start. I'm not quite sure for the first poster. Is Andrew, are you presenting?
Yes.
For that? Okay. I wasn't sure if anybody of your co-authors is going to be present. So for the first poster, I think we had some hype yesterday already. Some people were talking about it in the chat about that there was a poster about the entopins and which ones are the best. So there's a group from the Frost Museum at Penn State University, and we have the authors are Muhammad Ishak, Laura Potruas, Darren Pagan, and Andrew Deans. And Andrew is going to present this. And the title of the poster is "What are the best pins for entomotaxy?" And we're going to have just a couple more minutes before you can start.
Okay.
And if you want to share your screen with the poster, or if you have any PowerPoint, I don't know, you can do that now. And I think Nicole might have to give you permission to do so.
You should be able to share your screen.
Yes, and like Christy says in the chats, you can leave comments on the posters, and hopefully the authors then can go in later, and if you have questions, and answer them. You need your password to access the posters, and they will be up for, I think, quite a while after the meeting, and probably be archived in a past meetings page. So, whenever you're ready, Andrew.
All right, so can you see the poster?
Yes.
All right. Hi, and thanks for giving me this opportunity to introduce this poster. Last January, I put out a call to the ECN listserv to help us gather data regarding which, should I keep going? Okay. To help us gather data about which traits are important for insect pins, and in terms of style or brand, and then also to solicit donations of pins that we were going to put through a set of tests designed to explore those different traits. So we're very grateful for the donations that we did receive. We actually got a lot of pins from a lot of people and have over a dozen brands, I think, and also different types of pins represented. And I'm also happy to report that the experiments are underway, and we have some of our first results, which is what you see in this poster. We designed the initial research as an REU for an early stage engineering student, Mohammed Ishaq, who spent the summer learning the requisite engineering principles and designing the rig needed to perform this first set of tests, that is the tests on the shaft properties of these pins. And you can see from his poster that the test design wasn't exactly straightforward. It required lots of iterations of the jig design, because I think this lab actually is typically bending things like rebar or airplane wings or city buses or something. I mean, based on the equipment that I saw in their lab, it's like a pretty industrial and pretty awesome engineering lab. Anyway, so Mohammed was able to finalize this design and run a test on a subsample of our pins. And you can see some of the results in the upper right of this poster here. The x-axis reveals the displacement of the pin. So the pin is put in one of these jigs and put into, it's almost like a hydraulic press that measures the force applied. So the x-axis here is the displacement of the pin as force, which is the y-axis. is being applied. And you can see from this result here, which is the BioEquip #2 stainless steel pins, that that it approaches an asymptote, at which point it hits what's called the yield point. And at this point, the pin is permanently deformed. And then the performance of each of the pins is kind of unpredictable after that, after this point. Okay, so not all of our results are made it into the poster, so I'll give you a couple quick takeaways based on my reading of the data. One is that enamel pins tend to outperform stainless steel pins in this type of test. So you can kind of see down here on these bottom graphs, the stainless steel on the left and the black enamel on the right, although the performance wasn't that different between these two types of pins. But it gets me thinking, you know, if we're going to do a lot of pinning of heavily sclerotized insects like ironclad beetles or mutilids, that you might want to consider an enamel steel pin. That said, the performance of all the pins, except for Fusus, was not that different from each other. And I'm comfortable recommending at this point, based on this small set of tests, either the BioEquip or Endocrisis pins of either stainless steel or the enamel. Fusus pins, which you can see in this graph down here, this purple line, definitely did not perform. So Fusus were insect pins that we found on Amazon for sale, in which I saw one of my graduate students in my insect taxonomy class using for his collection. They are not performing at a level I would say is good for sort of long-term archival specimens. And then one more bullet point, one more result that's not actually represented on this poster is the inconsistency of the performance of Emil Artel pins, which were my favorite as a grad student, the elephant pins. You can see from this, these BioEquip pins that, you know, this is a test of 5 pins, and you can see that they're all kind of on top of each other, all performing in a similar way. The Emil Artel pins were all over the place, which suggests some sort of inconsistency during the manufacture process. So we have a lot more to explore regarding that performance. And not just our pin shaft performance, but we have other experiments planned for how much force it takes to pull the head off, and some corrosion experiments that we're going to design hopefully in the coming semester. So that's all I have for my quick introduction. I don't know if there are some questions that people want me to address while I'm here.
Please put your questions in the chat or raise your push the button, raise your arm. And then I have a question. So are you going to do any other experiments on like pinhead performance, or the tip on how fast they go dull or?
Yeah, so we haven't thought much about the pinpoint tests yet, but we do have ideas for the head performance. We're going to have this pull-out test, basically measuring how much force is required to pull the head off a pin. And we have pins of different styles. We have spun brass heads, we have nylon heads, or some other kind of epoxy head. So that'll give us a range of different materials to do these tests on. And we also have some ideas for corrosion test, which will involve some sort of salt spray. But we haven't made it that far in those, in the planning yet.
Okay. Brett Radcliffe wants to know, what about adhesion to the insect body? Are you going to test anything like that?
That's also a great question. I don't have anything planned yet, but that's certainly something that we could think about. In fact, you know, if anybody has any suggestions, we actually have a pretty big sample set of pins right now that we could do a diversity of experiments on with a number of replicates. So we have the head pull-out tests, we have these bending tests we're going to continue doing, and if anybody has creative ideas for other ones that you want to know, like adhesion, let me know.
Anybody else? There's a lot of people saying that they are looking forward to the corrosion test.
Yeah, me too.
Oh, Gene Hall says, if he may have missed that, but was there a mention of impacting points if using archival paper?
Yeah, so this was the set of experiments we haven't yet approached to think about, but it's definitely on our radar. And it's in our pin challenges section of this poster, where we acknowledge that the pin points can be problematic if their integrity is disrupted. So it's definitely something I'm interested in testing for these brands and styles.
Okay, Margaret there says that for the corrosion test, insect hemolymph might be important to test.
Yeah, so our initial ideas involve some sort of salt spray, which I think is what our corrosion experts here are recommending. And I definitely would like to explore different kinds of chemicals. And so it might be good to think about the chemistry of insect hemolymph or the chemistry of insect sort of soft tissues in general, and whether we could design not just a salt spray, but a spray that has certain kinds of lipids or acids or other kinds of chemicals that may interact with pins.
Okay, Denise, I can't read her last name on my screen, wants to know what if how heating pins up when removing damaged insects would
Could affect, yeah.
affect the pin, yeah.
Yeah, so Denise is remembering a paper that we read together as a lab group, looking at approaches that I think were used at the Natural History Museum in London for removing pins from specimens by heating them up. Yeah, that's actually something I hadn't considered in terms of a test of long-term performance of pins, but that's an interesting idea. Quality. Do I believe the quality has changed over time? Could stocks of older pins be better or worse? That is a really good question. We have a really small sample size that we're presenting in this poster. I mean, we really just looked at really just pins of 3 different brands of 2 styles. And I will say that the older pins, the Emil Artel, the elephant pins, did not perform as consistently as the newer Entocrisis or BioQuit pins. And I don't know if that's a factor of age or if manufacturing processes have become more consistent over time. It's a really good question and something I think we'll have a better answer for as we do increase our sample size outside of this small set. Do certain pins hold points for point mounting better than others? It's a great question. That could be another set of experiments that we design, and that would require some thinking about how to test those sorts of variables. And actually it'd be interesting also to test different types of cardstock or Bristol board or different paper types.
Well, thanks. I'm definitely looking forward for, I don't know, another poster or the paper or whatever you're working on. We have about 1 minute until the next poster, but if you have any more questions, put them in the chat and Andrew may be able to just answer them there. And otherwise, thanks for presenting this. It was
yeah, thank you. I'm also looking forward to the results. I'd love to be able to present sort of I would love to be able to answer the question that's in the title of this poster. What are the best pins for entomotaxy? So that's my aim. Maybe next year at this time, we'll have the answer.
Okay. Thank you.
Thanks.
All right. Next up is Luis Nastasi, and again, Andrew Dean from the Frost Museum. And Luis is going to talk about a survey of arthropod-associated objects in entomological collections. And you can share your screen, or I don't know what you have prepared.
Sure, let me see if I can share it. All right, is that coming through for everyone?
It's good on my side.
All right. Well, hello, everyone. Thank you for joining us for the poster session. My name is Louis Nastasi, and together with my advisor, Andrew Deems, who actually you just heard from about insect pins, we've put together an interesting preliminary investigation of what I'm calling arthropod-associated objects and their presence in global insect collections. So arthropod-associated objects is kind of an invented term that I came up with as kind of a broad swath of these objects that are present in entomological collections. I'm kind of loosely defining this as objects that are associated with arthropod biology that provide additional data, but they aren't exactly the archetypical arthropod themselves. So examples of this are things like galls, leaf mines, preserved egg cases and plant material, exuviae, and so on. These objects that are directly connected to arthropods, but may not be the exact specimens themselves. And these are important for a number of reasons. Taxonomy, for instance, these objects can provide characters that aren't otherwise present in just the arthropods themselves. They also provide connections to natural history, especially things like host organisms or interaction networks. And they're generally kind of in this awkward place in collections where they're not insects, but they're often kind of forced to conform to standards for curating and preserving insects in these collections. So to kind of gauge the presence of these objects in collections, we devised a 23-question survey that was distributed via Google Forms through email and social media. We collected responses from February 11th to October 6th of this year. Here's an example question, but if you see my poster, there's actually a QR code to the survey itself, which was closed until recently. I have reopened it in case any of you are feeling inspired by this to contribute additional data. So that link is on the poster itself. And our major objectives of this were, again, kind of preliminarily speaking, trying to determine the holdings of AAOs present across global collections, trying to identify very basic preliminary trends in curation and preservation of these objects, and trying to investigate the level of use of these holdings. We received 24 responses from different institutions from 5 countries, including the US, Guatemala, the Netherlands, Peru, and the United Kingdom. And in terms of the taxa represented, it should be no surprise to any of us which 5 orders were most reported. Out of the 12 orders that people reported in their collections, the most popular by far was Hymenoptera, primarily due to the presence of galls. The other big 4 orders made a large appearance, accompanied by Hemiptera as well. In terms of the actual AAOs that were reported, galls were again the most popular, followed by nests, pupae, leaf mines, and a variety of other objects comprising different properties. So we were able to draw 3 kind of general conclusions from this data, one being the AAOs are well represented in general collections. We found that the mean number of types, meaning galls, leaf mines, etc., was a little over 4 per collection. And in terms of orders represented, it was greater than 2 per collection, but our mean here of 2.8 was a bit skewed, as a number of the collections that we received responses from were incredibly specialized in their taxonomic focus, either just Hymenoptera, just Lepidoptera, just mantises, for instance. So that average number of orders there doesn't really represent the holistic approach of this system. One of our other general conclusions is that curation and preservation methods are currently consistently applied to these objects across collections. All the institutions we received responses from use drawers and unit trays as their primary curation methods, again, kind of representing this kind of conformity for these objects to traditional insect-based methods. And also these collections tended to use exclusively dry preservation methods for these materials. And our final conclusion is that AAO holdings are generally underutilized within the collections community. We saw that no object digitization is present in the average collection. 17 of our 24 responses indicated no digitized objects that met our description of AAOs existed in their data. And the average annual visitor for collections was less than 1. I generously rounded it up to 1 here. And we specifically mentioned pre-pandemic as the parameter for this. Obviously, things have not really been functioning naturally the last few years. So this number is almost certainly 0 for the last couple of years. But prior to that, it seems like at most 1, maybe 2 visitors attend most of these collections to look at AAO holdings. But in most cases, that number was actually 0. So my personal theory is this is simply a result of the taxonomic impediment. Basically, there are not enough people working on groups that are necessarily connected to AAOs in a strict sense, in a way that would warrant their continual study. I imagine that if there were more people working on galls, for instance, this number would go much higher. But in our current system, it does seem that there aren't too many people specifically interested in examining these objects. So I'm going to briefly mention what I've been calling showcases, these two really remarkable examinations of different collections in terms of their AAO holdings. These are both outlined in much more detail on the poster itself, but I want to highlight the AMNH Vespid Nest collection of Jim Carpenter, which is this really remarkable effort to basically build bespoke nest mounts, which are specific to each individual wasp nest. And they're basically these kind of Frankenstein foam sheets that hold these nest pieces together along with label data. And also the Ferris Mackenzie scale collection at UCDC, which is this really remarkable collection of preserved plant material and associated scale insects. So these are two really amazing instances of AAO collections with extensive holdings that cover either a wide taxonomic scope or have a really amazing preservation and curation methodology applied to them. So I'm going to wrap up my brief presentation introducing the topic here. A number of individuals were able to actually meet with me over Zoom as kind of an interview style to expand on their survey responses. So I'd like to thank all of those individuals. Also, Denise Montelongo and Laura Porturas provided some general feedback on survey drafts. And I would also like to acknowledge the National Science Foundation for supporting the underlying funding that went toward this project. I'm going to share the actual poster itself here quickly so everyone can see that simultaneously. All right, let me get this shared here. All right. Does anyone have any questions?
Thanks, Luis, for the presentation. It's a cool poster. Is there any collection that you got responses from that's specialized on exuviae? Or
so the general tone of a lot of the survey responses where I kind of cobbled together random things spread throughout a large collection. In terms of exuviae specifically, we didn't see any collection that had a specifically large holding. I can specifically mention one response, which is actually our response here from the Frost Museum, with a pretty extensive collection of dragonfly exuviae from the Beatty collection of Odonata. But other than that, my impression of exuviae specifically was they're kind of mixed in random drawers, randomly collected at times, things like cicadas and so on. So no collection seemed to be very concentrated on that specific area.
There's a couple of responses in the chat from the Academy in Philadelphia and the Harvard have a Odonata exuviae collection. Any other questions before we move on?
There's a question from Erica. Did most people indicate using the brace pins to keep things from breaking, or was it mostly just to keep things that were loose?
So in terms of the actual methods applied to these objects, a lot of people seem to actually be pinning a lot of these objects directly. For instance, a lot of images of galls on pins were submitted to me. Exuviae did seem to be pinned or preserved in ethanol as well. In terms of bracing things in place, from what we see, mostly would apply to galls, especially large galls, either on segments of branches or large leaves, where they wanted to maintain the whole kind of gestalt of the object. In that case, it does seem that pins were used to brace those in place, but it does seem like mounting was actually a preferred method, which I found very interesting. Again, another example of like conforming to insect-based standards for these associated objects.
I have a question for you, Lewis. Great poster and talk, by the way.
Thank you.
So this is a survey of arthropod-associated objects in entomological collections, but I feel so many of these could also be in herbariums. Have you considered like perhaps reaching out to, sharing your survey with them to see if they have some interesting insights?
So this was something that we were floating around in the very early stages of this project. I was even in contact with the Penn State Herbarium trying to see what exactly they would have thought. My general impression from my conversations with them is that because they're mostly botanically focused, if there are things like galls or leaf mines present in those collections, they're likely not something that's reported, and there's not really an easy way to access the presence of those objects. It might be entirely possible that they exist in those collections, but I'm skeptical as to whether or not there's really a way to assess that in a broad sense.
Cool, very, very cool. Maybe as we head into a future of extended specimen network, great, it could be a great opportunity for collaborations. Absolutely.
Well, thanks, Luis. Great presentation and great poster. We have to move on. The next poster is by Charles Farnham of the MZC at Harvard, and he's going to talk about SQL and Python methods for cleanup and standardization of locality and collection event data in MZC base. We have a video from Charles.
Oops, sorry, let me reshare. I don't think I got the optimized for video. Yep.
There's no sound.
That sound's not going through.
No.
Sorry about that. It should be. Try it again.
Hello, my name is Whit Farnham. Hello, my name is Whit Farnham, and I am the curator.
Still no sound? It's
There's
not.
sound.
There is?
Yep.
Okay, sorry.
Curatorial assistant at the Museum of Comparative Zoology. Today I'm going to talk about the processes and tools that we created to clean and merge our localities during the pandemic. So a locality is simply where on earth something was collected. The reason we want to clean them up is primarily to find mistakes that have drifted in over various data entry schemes, and make sure that everything collected at the same location is actually in the same location in the database as well. And it will make georeferencing easier later on by simply reducing duplicate efforts. Before you start your cleaning process, you want to set a standard for how you want to clean. More detail on this can be discussed later, but our basic process is start at the highest possible geography and go to the more specific geography. We have a separate field for everything like a county level or above, and that is controlled. And everything below that is kind of free entry string. But if you see with this example, you want to start with like the city and then move something more specific in the city as opposed to the other way around. So you're kind of narrowing in the farther you go into the description of the place. This process also involved us exporting large quantities of data from the database and working with it outside the database. MCZ Base is a web-based application, so fortunately this enables us to quickly reference and edit any record we want to using a URL and an identifying mark on an identifying number for the record. Once you have this, you can sort and filter the data as required to kind of find matching and similar things, update in the database, and then mark everything else as duplicate for deletion. And this is going to save you a lot of clicks on the back end by not having to go through repetitive tasks of deleting and merging, etc. Disadvantage of working outside the database is that there are no controls on stuff. So errors can creep in And if there are delays between what you have in the sheet and when you upload the database, you need to deal with those mismatches. So quality control steps need to be built into your process if you want it to go well. The basic workflow we employed during this was to export the data, make the edits and the markings for keeping and deletion as required, run the quality control process. If there are errors, fix the errors. When there are no errors, the programs will export a project-specific update file that is basically just a bunch of columns for matching purposes that will then go into the database administration for update, etc. So the first step of getting data out of the database is going to involve you sitting down with your database administrator and determining what fields you want in your query to determine which localities are the same and different. Once you have this, you can take the parts of the query and you can break them apart, and I have a program that enables you to then make larger queries using OR statements from that. So here we have a set of states. We have the portions of the query template that were given to us earlier, and then we can use that to generate a larger query with a lot more criteria. So you can just take out and manipulate large chunks of data at once without having to kind of constantly go back and forth with import and export. Once you have the data, we look at a cleaning example. So in this case, Coolidge Corner, which is in Brookline, Massachusetts, has been duplicated because they went with the more specific over the more general first. So this first one needs to be merged with the second one down here. And we do that by taking this locality ID here, copying and paste it into the parent locality here, which means that all the records attached to this locality will be attached to this locality, and then this locality will be deleted. If you'll also notice here, we need to add the county to the higher geography here and here. And then since Brookline is unique, it just gets an X because it's not going to be merged with anything, and that just makes filtering it out easier. Another important thing to note is that if you are just sorting the specific localities by alphabetical order, you will not be able to find the duplication here of the Coolidge Corner. To do that, you would need to be searching by small, 3 to 5 substring fragments to make that work. That just makes the bytes more manageable and easier to work with. Once you're ready for quality control, you're going to put it into an MCZ-based dataset tool that I created that has a set of general quality control functions, a generalized error reporting tool, and as you get into more specific projects, it can be expanded upon to return project-specific update reports. The most useful thing of this package is that it can be expanded and built upon to be more specific while kind of maintaining a core central set of useful tools that can be shared across different project required needs for this program. And I will discuss that with you, with anyone who wants to discuss that more after this. The increased flexibility means increased chances of error. So we need to deal with cases where a record is both going to be merged and is also designated as a parent, because that's not really viable. We need to deal with cases where what are supposed to be unique identifiers are appearing more than once. We need to deal with the empty fields where things are required, and we need to deal with something that is also assigned to more records than it can legally be assigned to. And then there are also more project-specific errors that also have to be dealt with, and those are on a case-by-case basis. This is a sample data export. It's just very simply which field has the error, what value is causing the error, and a specific problem that it got flagged for. So as a result of these works, we started with 61,000 locations. We ended with 42,000 locations, so we cut it by about a third. And most of these have been georeferenced at this point. And these tools enabled us to spend the efforts in merging determining which locations needed to be merged, cleaning up and researching them. And then the automated active merging could just be done to the back end. So our energies can actually be spent on the actual project, not the repetitive tasks. The next steps we want to take are to develop tools to merge duplicate collection events. So this is the next step where the localities are the when or the wheres, events are the whens attached to the wheres. So they're kind of the next level down. This is going to require us to create a clustering algorithm, a metric to measure how effective the clustering algorithm is. And ultimately, we're going to need to process 180,000+ of these events. So automating some of that grouping will be a useful time and energy for us. And ultimately, we want to be able to just quickly attach events to specimens through numbers or some other system so we don't create any more duplicates. I want to thank Crystal Myers and Rachel Hawkins-Sipe for just general advice and help in building these workflows. Toby, Patrick, and Hannah for the hours and hours spent merging localities. And Brendan Haley and Paul Morris, who actually gave me all of my SQL templates and actually did all the updating work on the back end as well. And I will take your questions now. Thank you very much.
Thanks. Do we have any questions for Charles? Margaret?
This is an interesting talk, and it's good to see automation of that kind of data correction that needs to be done in pretty much all databases at some point. I found it a little confusing, though, that you called the corrected, that you used the term parent locality for the corrected version of a locality, since parent usually is involved in a hierarchical relationship, which that is not. Something like corrected locality or correct locality would seem to be a little clearer. I would expect the parent of a locality to be, say, a county, if that's the next higher up level.
Yeah, that's a fair nomenclature difference I could have changed. I tend to view it as like I'm taking a bunch of things and kind of where they exist on like the tree of how they're going to flow up. And that's kind of the master thing. Everything, all the localities are kind of flow into that one. So I viewed it as a parent in that case from kind of structure node standpoint. So
Yeah.
that's cool that.
It's more of a sideways flow than an upward flow, though, really.
That's fair. Yeah.
Anyway, good to see you. I haven't seen you for a long time.
Tommy has a question. Does MCZ base have any tools now that keep the localities from being duplicated in the future, like string matching or so?
Not currently. We're kind of doing some stuff on our workflows to kind of keep that from happening, but there's nothing explicit that keeps them from being duplicated going forward, unfortunately. That's something we should probably address on the database end. Yeah. Which is not something I have access to. These are all tools I had to create for a database. I don't have that level of permission to deal with.
Okay.
To export, I don't have any import authority.
I think Karen has one more question, and after that we have to move on.
Yeah, sorry, I actually forgot what I was going to ask. But is this specifically Arctos, or can it be used in other platforms?
So it's built on Python, and I built the system to be generically flexible for CSV spreadsheets. So it's meant to be basically if you have a spreadsheet of data, you can use it on any spreadsheet of data.
Awesome.
Yep.
Thanks for the poster and the little video to explain it. And we got to move on because we're learning a little late. If you have any other questions, please put them in the chat. All right, next we have Jenna Endek and Christy McCain from Colorado. And they're going to talk, the poster is about JARD potential, a nationwide survey of fluid preserved entomology collections and management best practices.
All right. Hello, everyone. I'm Genevieve, or Jenna. I'm currently a second-year master's student in the University of Colorado Boulder Museum and Field Studies program. And I have become really interested in the use of fluid preservation and entomology collections. As we all know, pinning is usually the main way that we store and preserve our insect specimens. However, fluid preservation offers a lot of benefits and is also used as well. It's really good for aquatic taxa as well as immatures, and it provides fluid support and can also act as a fixative as well as a continuing preservative agent with fluids such as ethanol. Fluid preservation practices have been pretty well established for things like vertebrates and non-insect invertebrates specifically. The big manual that everyone in vertebrates loves is Fluid Preservation by John E. Simmons. However, I've kind of become interested in the active management of fluid collections because over time with fluids, especially like ethanol, what happens is that when you have a solution, for example, 70% ethanol, which you create by mixing deionized water with pure ethanol, what will happen is that the ethanol actually has a higher vapor pressure than the water, and so will evaporate out of solution faster than the water. And so oftentimes if you see in your vials, if it starts evaporating over time, not only is the fluid getting lower and possibly making so that specimens are at risk of desiccation, but it also is actively lowering the concentration of the solution. So even if you already put 70% in there at the beginning, it might now be 50% or lower, and 50% or lower is actually no longer a biocide. It's no longer preservative. So that's when things start to grow. So topping off is really what I'm interested in, and that's refilling the bottle. And yeah, and fluid preservation is also really interesting because it can serve as almost part of the extended specimen. People can now sequence DNA and identify taxa, for example, in large bulk samples just by sequencing the ethanol, and also ethanol and other fluid preservatives. Depending on the exact preservative, can be good for preserving DNA in the specimen itself. So for example, here I have this jar of lizards because I'm currently working in the vertebrate collection. So what we do is when we have to top something off, first we have to check the concentration. And usually checking concentration in the past has been very difficult for collections because especially collections in entomology where the vials are smaller, because it requires a certain amount of the liquid to put, say, a traditional hydrometer, which is a floating glass rod in. So here, what the collections manager and my collection and the vertebrate collection here has done, Emily Breaker, is she has bought this meter for our collection. And this is a digital density hydrometer meter. And these run in the range of about a couple $1,000s. I think this might have been $5,000, but they work really well because you can immediately just suck the ethanol up into the meter. And there, we've got 70%. And so this is really a great tool. However, I think it's more difficult to take these methods and put them in entomology collections because of the smaller nature of specimens. And also specimens may be more likely to lose an antenna or some tarsomeres, and you can't get that sucked up into the meter because it can break the delicate tubing inside. So basically, this poster is me introducing my thesis, which is going to be an assessment of the best practices for topping off and maintenance of fluid preserved specimens, not only ethanol preserved, but other fluids in the US. So I would like to determine the best practices for managing these fluid collections by gathering expert opinions. And that would be from interviewing curators and collection managers that have expertise in taxa, say, Odonata, where they have a life stage that is water obligate, and their methods that they've learned over their careers for managing their fluid preserved specimens. And then I'm also going to do a survey of our material here in our museum of a bunch of fluid preserved specimens over time to see how they've been impacted by the overall storage state of the collection. What kind of caps are on the bottles? What kind of bottles are they in? How often are they topped off? And so I'm also going to do a little experiment to look at that as well here. But then the biggest thing for me presenting this poster is I would love to do a survey of all the entomology collections in the US to see like what y'all's procedures are for managing your fluid preserved specimens, and then just get a sense of how much material there is out there. Because I know when I've worked at insect collections, of course, it's all pinned, but you do have fluid preserved specimens as well. And maybe those are a strength of the collection, and maybe those are something off to the side that aren't as actively managed or maybe not as actively used in in collections or in research. So basically, please fill out my survey. I'm going to put a link in the comment here in a second in the comments. And then also, I would love for folks to contact me with information on people that I can interview to gather their expert opinions on, as well as just like other knowledge to share for my thesis. And then I would ultimately like to publish this in a collections management journal, and as well, so I can inform the community on the best practices for managing fluid preserved specimens. So yeah, thank you so much. I will put the link in the comments here. Hope I explained myself well.
Thanks. I think you did, and it's an interesting topic, and I think some people in the chat said that it's ironic how expensive some of these tools are that we should use, but we can't afford them because of funding. Any questions for Jenna?
I've got one. Can I just speak up?
Yeah.
Again, this is a great topic. I'm really glad someone's looking into this. One question I had that I'm kind of hoping you might look into is to what extent people should fill up actual vials themselves, whether, because I've heard two different opinions about this. One of them from a collections perspective is that you should never fill it all the way to the top because of changing vapor pressure. And if you fill it all the way to the top, the specimen's going to, or the vials can explode just because of changing vapor pressure. But I've also heard from people in the field that if you're using glass vials in the field, you need to fill it all the way to the top because air bubbles can destroy specimens if you're transporting them. So it's something I've heard both ways, and I'm curious like what the trade-offs are and what the best practices are in that direction.
So in our collection here, of course, we have, I'm in vertebrates right now, but love bugs and vertebrates as well. So for us, we always try and fill things all the way up to the top because from the collections management standpoint, that allows you to know that if you filled everything up to the top, you can walk around and then easily see which things need to be topped off or have the concentration measured to see if you need to fill them up with a different concentration of fluid. So it kind of gives you that starting point where you can be like, oh, okay, this one's declined this much over time. However, with the smaller vials that entomology collections often use, I've totally done the thing where I filled it up too much and then the bottom falls out and you get bugs all over yourself. So that is something I'm also considering for my project, and is why I would love to interview experts to get their opinions on these things, because you can kind of go either way. It's a give and a take, what works best for your collection and your purposes.
And it might be interesting to do some experiments to see like how much air bubbles affect fragile specimens. If you can get some, what's it called? Some weight, I don't know about waste, but like useless fragile specimens, and actually like look at how much an air bubble moving around in a vial would actually mess up something like a trichopteran or something that has lots of little tiny parts that would fall off really easily.
Yeah, that's a really great idea. And hopefully something I can get to in the timeline of my thesis.
Margaret?
Yeah, there was an interesting talk, and there's lots of, I'm sure you'll be learning lots of interesting things in the course of your work. As far as the air in the vials, what we've typically done at the Field Museum is get rid of all the air, and not just by filling the vial, but by filling it with a little bit of an air bubble, and then you stick something thin like a pin or a little nylon tag hanger from clothing, stick that in beside the stopper, and then stick that in so that the air all comes out. Then you don't have any vapor pressure issues because there's no place for there to be any vapor. And I think that also if you don't fill them and leave them, say that like if you did the equivalent to a vial of what you've got in that jar beside you, then if you have temperature fluctuations, you will have pressure changes and things, which, I mean, in the very extreme case could blow the stopper out or something. But if you eliminate the air, that's not a problem. And then you also don't have the chance of air bubbles being in there to damage specimens.
Interesting. Yeah, that's a really cool method. Thank you for sharing.
Yeah.
Thanks, Jenna. It was a really cool poster. I really liked it. And but we do have to move on. We have about 10 minutes, I think, till the general meeting. So everyone can get some refreshments for the meeting. And then Chris will take over at 15 after the hour with the general meeting.
Thank you, Oliver. Yes, our business meeting. So please come back for the business meeting. We'll start in a few minutes. Let's take a break. Thank you.